Good morning, students. As you all know, that our country is facing a crisis because of lockdown. So, our management, our principal man, our teachers have decided to conduct online classes so that we can reduce our losses in terms of studies. So, let's have a quick recap of what we have done earlier. We started with the second chapter, that is molecular basis of inheritance, and in that part, we have done DNA, the characterization of DNA. Then we've done the packaging of the DNA, about the Watson tree DNA model. So, now today we are starting with the search for genetic material. That is the next topic. So, in the search of genetic material, first of all, you all know that nuclein was discovered by Miescher, and after that, various discoveries have been done. Let's say about the Mendel's part that we studied, about the Walter and certain Boveri and certain theory of discoveries, and then we have done the Th. Morgan studies. So, after that, it was in 1928 that Frederick Griffith, in the year 1928, he performed a series of experiments by taking two organisms. His experimental organisms were, he took two organisms. One was bacteria, and second was the mice. Now, as far we talk about the bacteria part, he took bacteria that causes pneumonia. That is Diplococcus pneumonia. Now, in this bacteria part, he has two different strains. Now, you can also take Diplococcus pneumonia or Streptococcus pneumonia. Both are coccus bacteria and both causes pneumonia. So, in this bacteria, he has got two strains. One was R2 and second was S3. The R2 strain is a non-virulent strain, and the S3 strain is a virulent strain. So when I say the non-virulent strain, that means it is non-disease causing. And when I say the virulent strain, so that means it is disease causing. So what he did in his experiments, he injected R2 into mice, and the mice survived. The mice did not die. In the second experiment, he injected S3 into mice, and the mice died. So there was something within this S3 strain that killed the mice. That principle or that molecule was absent in case of mice, in case of R2 strain. So what he did in the second series of experiments, he then again took S3 and heat killed it. Heat killed, and again he injected into mice. So in this case, the mice survived. So this means when we call heat killed, so that means the organism's structure is destroyed, but the DNA as a molecule gets denatured. Denaturing means a double-stranded DNA converts into a single-stranded DNA. So in that case, the DNA remains intact. The property of the DNA is not lost, although the double-stranded DNA converts into a single-stranded DNA. And he again injected into mice, and the mice survived this time. So what he found, there was something with the S strain that again and again killed the mice, but when he killed it, the mice survived. So in the fourth part, what he did, he took the live R2 strain plus S3, again he killed, and again he injected into mice. This time the mice again died. So what was happened actually, the S3 strain has an outermost mucus polysaccharide layer which the R2 strain does not have. So when in the last part, he found it out that something was the transforming principle that converted the R2 strain, which was the non-virulent one, it converted into the virulent one. Although this was heat killed, still the mice was died. The reason for this is there was something from the S3 that transformed the R2. Hence, he gave this experiment as the transforming principle experiment. So from this experiment, he was not able to prove that what is the actually the hereditary material. He was just able to identify that something was there that killed the mice because of the R2 strain. So he finally gave the conclusion that some transforming principle from the S3 transformed the R2 strain and into the S3 one, and finally the mice died. Now to prove his experiments, there was other three scientists, that is Avery, McLeod and McCarty. They did the chemical characterization of transforming principle. Now, what is the chemical characterization? They actually found it out. Okay, what is the chemical that actually killed the mice? Earlier, it was believed that it was the protein that killed the mice, but later on, after their series of experimentations done by three scientists, that is Avery, McLeod, and McCarty, what they did is they first took the S3 strain, and out of this, they took three molecules. First was protein, second was RNA, and third was DNA. All these three molecules, they put it in a culture that contains R2. In this part, when they took the protein of S3, there was no transformation. Again, in this part, there was no transformation. But in the third one, when they took the DNA of S3, there was transformation. And when I say transformation, so that simply means the R2 gets converted into S3 and kill the mice. So with this experiment, they was almost able to prove that DNA was the genetic material, but still they also performed one more confirmation experiment, in which they took the DNA of S3 and used various enzymes that digest the various macromolecules present in the cell. So first, they took the enzyme proteases. Now you have all studied in chapter in 11th in chapter 9 that proteases digest proteins. So in this case, when S3 DNA of S3 was digested with the help of proteases, the DNA didn't get digested, and hence the transformation took place. Second, they took RNAs. Again, the transformation took place. And the last part, they took DNAs. And this time, there was no transformation. So this finally concluded that DNA is the only material that caused the transformation from R2 to S3, and hence the mice finally died. So finally, with their series of experiments, these three scientists, that is Avery, McLeod, and McCarty, was able to prove that DNA is the material. But again, at this point of time, it was proven that DNA is the genetic material, but it was not proven that DNA is the hereditary material. So there are continuous series of experimentation after this experiment also, that was the final unequivocal proof to prove that DNA is the genetic material. So we'll conduct the second experiment, the second lecture. Now please go through this experiment from NCRT, and I will be sending the questions to you for MCQ questions to the group. And for the fifth chapter that we already completed, the first chapter one, before that the principles of inheritance and variation, please solve these questions. Thank you. Stay home and stay safe. Thank you.